Well, welcome everybody. I'm Dr. Chris Nowinski, co-founder and CEO of the Concussion Legacy Foundation. We're very honored to have a special guest here today joining us, Dr. Ann McKee. She is Chief of Neuropathology at the VA Boston Healthcare System, Director of the Boston University CTE Center, and also Director of the VA-BU-CLF Brain Bank. She's also a professor at Boston University. She is the world's expert in chronic traumatic encephalopathy research. Known her now for 12 years. We've been very proud to be a collaborator on the Brain Bank. 12 years ago, I met Dr. McKee and I said, Dr. McKee, I think there's this really big CT problem, which she was already aware of. And I said, we need to accelerate the research on this. And can we build up a brain bank with you? And she already led multiple other brain banks. She was an expert in many neurodegenerative diseases, but she made the commitment there to say, yes, I will make CTE my life's work. And she has done some amazing things with it. And so what we're here to talk about today is her latest research study, which is the largest ever study on CTE cases, over 350 cases, to help us understand CTE progression. So Dr. McKee, thank you so much for joining us, and I will yield you the floor, and then everyone knows we'll have Q&A at the end, so feel free to write your questions into the Q&A as they come to mind. Dr. McKee, the floor is yours.
Thank you, Chris. Yes, I've had the honor of knowing you for 12 years, and it's been a wild, wild ride. And I'm happy to share some of our results with the audience. This is a paper I'm very proud of, and I'm going to tell you about it. It was published recently in the journal, or the Journal of Acta Neuropathologica, which is a very prestigious journal for neuropathologists. And the title of our study is Characterizing Tau Deposition in CTE, the Utility of the McKee CTE Staging Scheme. And my co-authors, Mike Alaskow, John Cherry, Russ Huber, Yorgos Tripodis, Zach Balcom, Neil Cowell, Lee Goldstein, Doug Katz, Bridget Dwyer, Dan Dantrevere, Joe Palmisano, Brett Martin, Robert Cantu, Robert Stern, Victor Alvarez, Jesse Mez, Torstein, and myself. And so let me just tell you a little bit about why we wrote this study. My slides aren't. Okay. So back in 2013, after I'd met Chris 5 years earlier, and we'd started this brain bank, we started collecting brains of individuals who were subsequently diagnosed with CTE. And by 2013, we had 68 subjects with CTE. And that was actually already 4 times more than any other cohort. No one had ever had this kind of experience with CTE. And I remember Bob Cantu was really pushing me to write this paper on the clinical pathologic characteristics of CTE. And I was hesitating a bit because I wanted to make sure that I really understood the subject. I felt that there was a lot of pressure. We were under a lot of scrutiny. There were a lot of skeptics at that time and still are. So I was hesitant. And even though Bob really wanted me to publish, I wanted to make sure that we had things right before we went to press. And what we did in this study back in 2013 is we proposed for the first time criteria to make the diagnosis of CTE by pathologic examination, and that had never been done before. That went on to be refined and adopted by the NIH, but we also, in this study, proposed a staging scheme for pathologic severity. And let me just tell you a little bit about the mechanics of that. So this is an observational study, and a lot of science is observational, and some of the greatest advances in science have been observational. And this was a classically observational study. And what we had done is, as we were collecting the brains of these individuals with CTE, I was looking at them and I noticed a pattern of pathology. I noticed that the individuals who had the least amount of pathology tended to have lesions in this part of the brain. And as they had more severe pathology, it tended to spread out and involve other regions of the brain in what turned out to be a fairly predictable pattern. And I think I was pretty clear by the time we got to be about 35, 40 cases. It was pretty clear that there was a stereotype pattern of involvement in this disease from least severe to most severe. But I kept, I was nervous. I was nervous because I knew that we would be heavily scrutinized for this work. And so I kept gathering more cases came in. And finally, for unclear reasons, when we got to 68 subjects, I felt like, okay, I see the pattern, it's predictable, we can go to press with this. And so that's how we developed this staging scheme for pathologic severity. It was just observation of looking at many cases over those 5 years and finally coming up with what we thought was a predictable pattern of involvement. So the criteria that I mentioned, the criteria for diagnosis, went on to be refined and adopted by the NIH. There was a panel of expert neuropathologists. They looked at the criteria that we had proposed. They adopted it. In fact, they even defined what we call now a pathognomonic lesion. And they defined that pathognomonic lesion as an abnormal collection of tau around blood vessels, in neurons, astrocytes, and cell processes, at the depths of the cell site in an irregular pattern. And those were features of the disease that we had described in that paper, particularly the perivascular nature of the disease and the fact that it tended to be worst at the depths of the cell site. But we also presented in that paper this staging scheme that I've been talking about. And what happened with that was in the mildest cases, the cases that had the least amount of tau pathology, we could see that there were just 1 or 2 isolated lesions, generally in the cerebral cortex. In fact, most of the time in the frontal cortex, and very specifically, it tended to be superior and sometimes superior lateral in the frontal cortex. But that was all there was in some of those cases. And then we noticed the cases that were just a little bit more involved, where they had just a little bit more disease, they tended to have slightly bigger lesions, and the lesions involved multiple lobes of the brain. So now it wasn't just in the frontal, it tended to be in parietal and maybe temporal as well. We called that stage 2. And then stage 3 was very different than the previous stages because we started to see involvement of areas of the brain that are extremely important in memory and learning. And that's the hippocampus, also the entorhinal cortex, and the amygdala. And the amygdala is considered the seat of emotion in the brain. It's thought to regulate impulsive actions. It has a lot to do with aggression. So we were seeing a different disease in these individuals. When they had widespread lesions, the lesions were bigger, they started to have involvement of the medial temporal lobe, those important areas of the brain that hadn't previously been involved. We called that stage 3. And then stage 4, we saw disease everywhere in the brain. It was really quite severe. It affected most of the cerebral cortex. It was very severe in the medial temporal lobe. And we called that the most severe stage, stage 4. You can see that the age of the person at death correlated with the stage, and that the youngest people tended to die with early stage disease, and the older people tended to have more severe disease. And this is just a high power view, or a closer look at what these lesions look like. This is that stage 1 disease that we often see in young adults, 20s and 30s. The changes surround a blood vessel in the cortex. Here's that blood vessel. And you can just see this constellation of tau in what we call neurofibrillary tangles surrounding that little blood vessel. That's the earliest stage. And then stage 2, that lesion gets bigger, more neurofibrillary tangles, there's more pathology, and again, centered on that blood vessel. By the time you get to stage 3, where we're seeing the medial temporal lobe involvement, that is looking like a nasty lesion. You can see it's large. It's starting to involve astrocytes. It looks like it's coalescing or organizing. It's a well-established lesion. And by stage 4, it's actually quite a substantial lesion. There's a lot of astrocytes or scar-forming cells in the brain. And it's really quite large and affects very large regions of the brain. So we've used this staging system in many papers. Probably most notably, we used it in the JAMA paper in 2017, where we described 110 out of 111 former NFL players with CTE. And we've used it in other papers as well. Other groups have used the staging scheme, but it has never really been verified. It was always that observational study, and we'd never actually done any deep dive into how much tau is in the brain, and did the actual quantity of tau correlate with that same staging scheme that we described. And so that's what we did in this paper. We tested the utility of the staging scheme against very comprehensive, laboriously collected quantitative assessments of tau. So we used the brains of 366 individuals, so a huge number of cases. No one's ever looked at that many people with CTE. We looked at what we call semi-quantitative assessments of CTE, of AT8 or tau pathology. When we diagnose the case, we go through a very long list of areas of the brain, and the neuropathologist, myself or my colleagues, will rate the amount of tau pathology we see under the microscope as either 1+, meaning just a little, 2+, meaning moderate amounts, or 3+, meaning a lot. And we go through that in every region of the brain, and we have a large database that collects that material. So we use those semi-quantitative assessments, which we gathered over the last 12 years in those 366 cases, and we looked at that data compared to the staging scheme. We also looked at direct counts, absolute counts of tau pathology, and we used a computerized scan to do that. We digitally imaged the slide and then used computerized scanning to actually quantitate the number of pixels that were positive for tau. And that gave us a quantitative evaluation of tau. So a lot of very laboriously collected hard data on the tau pathology. We also looked at age at death, dementia status, and the number of years of play of American football. This is just a look at that computerized scan that I talked about. This is what we see as a computerized image of a glass slide that we look under the microscope. This is actually a computer photograph. It's extremely high resolution. You can make out the individual cells. And this machine is able to look at this area that we've delineated and actually come up with a count of how much tau is in that particular part of the brain in that particular person. And we use these quantitative evaluations to compare it to the staging scheme. This is just a graph or a table of the participants. You can see that they had a wide range of age at death, 17-year-olds, 100-year-olds. The mean age was about 61. All of them were male. Most of them were white. And most of them, 304, were American football players. We did have some, 90 with a military history, about 25%. Many played professional sports, but you can see we also had high school sports, college, semi-professional. And then this was the pathology results. I'm not going to go through this very complicated table, but just showing you out of the 366 cases we had about 60 cases of stage 1, 80 cases of stage 2, and the rest were the higher stages, stage 3 and 4. So the first thing we did was we looked at those semi-quantitative counts, and we checked to see if they correlated with the stages of CTE, and we found that it was statistically significant in every single brain region we looked, all 12 brain regions. Those semi-quantitative counts that we'd collected over 12 years were very significantly associated with the stages of CTE, all with p-values of less than 0.001. We also looked at that quantitation, the hard data, the hard numbers collected by that computerized scan, and we found that those were statistically significant across all the brain regions we assessed. And that was frontal cortex, hippocampus, subiculum, and locus coeruleus. And again, you can see that there was more tau in absolute hard counts in stage 4 compared to stage 3, compared to stage 2, compared to stage 1. So again, validating our staging scheme. When we looked at age at death, we found that the nature, the severity, and the distribution of CTE pathology followed an age-dependent progression. In other words, it gets worse as these individuals survive. Most people stop playing football or leave the military service, usually by, depending on the sport, 40, 50, but they leave the sport, and even though they stop experiencing repetitive head trauma, the disease continues to progress. It gets worse and worse as the individual ages. And this shows that most people that are older when they die have high stage CTE. Those that are middle-aged tend to have stage 3. And the younger groups, the 20 and the 30-year-olds, tend to have low stage CTE. But very importantly, some people with mild CTE are older. And that suggests that not the majority, but sometimes CTE is very slowly progressive, or perhaps not progressive in the minority of individuals with CTE, that some people seem to be resilient to the progression of CTE, in that they most likely stopped experiencing contact sports or military injuries at around the same time, but for some reason their disease didn't progress. But that, again, that's the minority. Most of the individuals, the disease gets worse as a person survives. And then this is just a heat map showing you that in all the regions of the brain that we looked at, frontal cortex, inferior frontal cortex, temporal, parietal cortex, if you look at the counts of tau, those tau counts go up in every single area as you get older. Looking at 20-year-olds compared to 80-year-olds, in every single line, this disease gets more severe over time in every region. And if we looked at just the demented folks, there were about 216 of the 366, 60%, who were determined to be demented at the time of death. And we found using binary logistic regression and controlling for age, that higher stage was associated with increased odds for having dementia, with an odds ratio of 1.64 for every stage. And that remained after controlling for all comorbidities, including vascular diseases such as strokes and other neurodegenerative diseases such as Alzheimer's, Lewy body, frontotemporal degeneration. And then lastly, we confirmed what we had found earlier in our 2013 paper. We found that the stages of CTE were significantly associated with years of football, and that more years of American football play was associated with increased odds for having a higher stage of CTE with an odds ratio of 1.1, controlling for age at death. And then this was something that we did at the end that I really wasn't anticipating, but we did a cluster analysis. That is, we sort of looked to see if there were any patterns or clustering of pathology. And we did some statistics. This is called a gap statistic. We looked at the optimal number of clusters. If you just sort of throw all the tau pathology into a big basket, is there any pattern or clustering of the pathology? And it showed us that there was an optimal number of clusters of 5. And when we looked at these clusters, and this has nothing to do with staging, nothing to do with counts, it's just clusters of pathology, we found that There were 5 clusters, and in the first cluster, the people with the least amount of disease, we saw involvement of the frontal cortex and locus coeruleus. So this really maps well with what our stage 1 disease is. So cluster 1 in this very different analysis looks like stage 1. And then we have cluster 2, where we start to see more pathology in more regions of the brain, including locus coeruleus, but it's fairly mild, but it's spread out to different regions. And that's a cluster number 2, and it correlates very well with stage 2. Then we get cluster 3, where we start seeing more severe pathology affecting more regions, again, cortical, basal ganglia, brainstem. Cluster 3 maps on to stage 3 very well. Interestingly, when we get to stage 4 disease and cluster 4 and 5, it appears that there are 2 groups, which we hadn't appreciated. There's one group that shows very severe pathology throughout the brain, and then there's another group, cluster 4, which shows more severe cortical pathology, but not as severe pathology in the hippocampus and in regions in the subcortical nuclei. So cluster 4 and 5 correlate with stage 4, but we hadn't realized there are actually 2 different patterns in stage 4. So what are the conclusions from this study? These findings support the usefulness of the CTE staging scheme to assess CTE pathology severity, and they support their continued use in the study of CTE. The findings demonstrate the distinctive regional distribution of tau pathology in CTE, as well as link this tau pathology with older age, dementia, and greater years of American football play. What we don't know as much about yet, but we're hoping to know in the very near future, is what are the clinical correlates of CTE across these different stages of disease? Are they clearly different? Can we identify these different stages of pathology based on just the clinical symptoms? We're not there yet, but that's something we are looking to do. And we also need to identify risk factors for CTE other than exposure to head trauma. We know that some genetic variables change your vulnerability to CTE. Some make you less susceptible, some make you more susceptible. We know that there are probably other factors that we still don't appreciate that play a role as well. So we still need to look at the risk factors that enhance CTE susceptibility and course progression. And so lastly, what do I think is the importance of this work? Well, I think that this work firmly establishes with hard data, hard quantitative counts established by computerized imaging, that we know exactly where CTE starts in the brain and where it goes and how it progresses with age. And these advances are important because there's such an emphasis on diagnosis during life. This will show us the roadmap of how this disease affects the brain so that when we use tau imaging to make a diagnosis of CTE during life, we can see if the imaging results overlap what we know is the pathology. And then these advances will also help guide us in the development of treatment strategy aimed at tau, aimed at preventing the spread of tau throughout the brain, and aimed at halting the progression of this disease. So I want to thank my collaborators. This is just some of them because it takes a massive team. I want to thank Boston University Alzheimer's and CTE Center and all the research staff, CLF. I want to thank VA Boston Healthcare System and all our many funders. They've been extremely critical for this work. Thank you very much.
That was incredible. I do want to thank your entire team for doing that. I have some appreciation for how much work went into that study, and it's incredible. I was looking at some of the chat. I do want to thank, I know some of our brain donor families who are watching, so we want to give you another thank you for allowing us to do this work by donating the brain of a loved one. I see a lot of folks who pledged their brain to us. Randy and Ray and some of our NFL guys and some other folks. So thank you guys for supporting this work in the future, although hopefully we never end up with your brain. We solved this problem before then. And I also have some caregivers here, so I want to thank you guys for joining. And a lot of these questions, really come down to how does CT affect the living? And so I've got about 150 questions that have been sent to me. I'm going to try to give you some of the ones we can answer quickly here. So let me just start with some that were sent in advance here, to include, and again, we know where your expertise is with the brain bank, but you're also an expert on everything else regarding CT. So how do we talk about treatment for CT right now?
Yeah, well, I think we've clearly established that tau is a major factor in this disease, and we know that it correlates with dementia. And the thing is, there are quite a number of trials right now for anti-tau therapeutics. They're mainly being used in Alzheimer's disease, because that's a disease that we have great biomarkers for. We're able to monitor the disease through imaging, tau imaging, as well as blood and CSF biomarkers. And those are being used in Alzheimer's disease. We know the tau in CTE is just a little bit different than Alzheimer's disease tau, but we're hoping once we are able to get a biomarker that really detects this disease, that we can use all those different anti-tau therapeutics, which are already in the pipeline, on individuals with suspected CTE.
Thank you. And in terms of how we talk about when we'll be able to, we think we might be able to diagnose CT in life, we're basically still at if things go really right, maybe 5 to 10 years, but we have to do a lot of work and get lucky.
Yeah, but I mean, just over the last couple of years, things have gotten so much better. We're pretty, I think people are comfortable with the biomarkers for Alzheimer's disease. The tau PET scans called fluorotaucipir work for Alzheimer's disease. They characterize the disease during life. We've got a lot of labs looking to develop great markers for CTE tau, which is just a little different than Alzheimer's tau. And there are multiple labs in multiple countries looking for this. And, I have to say, I'm very excited about the progress. And I think, with a lot of hands on deck, with a lot of financial support, we ought to be there at least within 5 years.
That would be great. The question about how CT might develop in cases of abuse. So I know you're trying to understand how different exposures to trauma might lead to different types of CT pathology, but would the answer be that we don't have enough cases outside of sports right now to understand abuse or military, and we need those?
Yeah, we definitely need, we want people to live long, healthy lives. But if tragedy strikes, we desperately need women's brains. We desperately need military brains. We desperately need abuse, physical, domestic abuse, because it takes numbers to really understand that. We can't do genetic studies without numbers. Right now, we're almost to 1,000 brains in the brain bank, something like 960. But genetics needs thousands. So we still need to get more brain donors, especially from military, women, and those different types of trauma, like physical abuse.
Now, there's a couple questions on related diseases. And so could you speak to what you find in terms of comorbid neurodegenerative diseases, and also what you've learned about the interaction or potential interaction between trauma and Parkinsonian symptoms and Lewy body disease or Alzheimer's? And I will say, I noticed in your paper that was interesting is most, the early stages, most people don't have comorbid diseases. But for example, stage 4, I believe about a third also had Alzheimer's disease. Right. But that's still relatively easy to differentiate what you're looking at with Alzheimer's and CTE, which is another question.
Yeah.
Correct? Yeah.
No, it gets a little tricky. It gets tricky when you've got a lot of disease. It's hard to see the individual trees in the forest because you've just got all of this disease. But it's a tenet of neurodegeneration in general, that once you get one neurodegeneration, you're more likely to get another. It lowers your resistance to everything. So people with Alzheimer's, you know, they have a higher risk for Parkinson's, and people with CTE also get these comorbidities, probably a bit younger than they would if they didn't have the CTE. Interestingly, in our work with the Framingham study, we found that repetitive head trauma through contact sports, also in addition to CTE, we also see increased risk for Parkinson's and Lewy body disease. So even without CTE, just that repetitive head trauma seems to lower the bar for getting Parkinson's related disorders. And in, you know, just anecdotally, in some of our very young individuals with CTE, people who have died in their 20s, we actually have a few who also have comorbid Parkinson's. So there is an overlap there. What else did you want me to talk about?
No, I think that's sufficient. Thank you. There's a lot of questions to get through, so I'll sort of work quickly, and I'll cut some short where I need to. There was a question, and this came up a few times from different people. This one is a longtime football player who's now 70 and asking, what should I expect going forward? And I thought it was very interesting, and this is always a very important question. I thought it was very interesting that you highlighted that you had a lot of a surprising number of early stage older people, and the fact that some people appear to be resilient. And so, you know, this is a time where we like to talk about hope, right? That we can't predict what's going to happen to people, and some people aren't going to be badly affected, although unfortunately a lot are, right?
Right. And not everyone is affected. I mean, we've done these other papers with risk, you know, exposure and risk, and we found that the number of years of playing football dramatically increases your risk, but it doesn't mean that everyone who plays football will get CTE. And certainly some people get it, and it doesn't get much more severe over time. So we are really focusing on those individuals because they demonstrate resilience. Now that may be partly mediated by genetics. They may have a different reaction to the injury. It might be inflammatory markers that are able to sort of wall off the injury or prevent the progression of disease. It might be other things, but we're very interested in determining that vulnerability threshold. What changes that for different people? There's a lot of hope out there, and this is not a, you know, so every case is different. It doesn't mean you're going to just progress and become, you know, just terribly affected as you age.
And one of the interesting variables that you've taught me about is that this paper is about tau, and tau is what's required for the diagnosis and staging of CT. But you've also identified a lot of other things that are going wrong in the brain and are different in every case, like TDP-43 and others. Can you speak to some of the other abnormalities we're finding with CT in the brain?
Sure. Well, you know, we do see TDP-43 in the brain. That's another neurodegenerative protein. We're still struggling with exactly what that means. Does it contribute to the clinical manifestations of the disease, the dementia, the behavioral, the mood changes? We're not sure. It's very complicated. You know, the more you get into anything, it gets very complicated. But the other thing that I think we're really interested in addition to neurodegenerative markers, we're really interested in the white matter and changes in the white matter, evidence of axonal damage, changes in the connections of the brain. And that may be something that stimulates some of the symptoms, even some of the early symptoms, maybe even in the absence of tau. So that's actually where we're spending a lot of time right now, is looking at those connections in the white matter. And can we say anything about those? Can we identify those in young people before they've gotten to the point where they have CTE?
So our friend Renato from Brazil, who we both know, wants to know, what do you believe is causing progression? Can you speak to the work you've done with Stan Prusner on understanding how CTE is spreading?
Well, we definitely think that there is seeding of the brain and spread of of the protein in the brain, that this tau protein that's inside a nerve cell can actually transmit, if it's in contact with a normal nerve cell, it can actually give that normal nerve cell some of this abnormal tau protein. And in that way, it spreads the disease from bad areas of the brain to otherwise, good areas over time. There's definitely evidence that tau spreads, that it can seed otherwise normal neurons. But we also think there's other things, primarily this inflammation that I talked about. There's a tremendous inflammatory response after head trauma, even the little hits that you think are inconsequential, those subconcussive hits that don't cause any symptoms. We see after those hits tremendous inflammation, and in CTE that inflammation gets worse, and as each stage gets worse, that inflammation gets worse. So could we put that inflammation in check? Could we settle it down? Could we treat that inflammation, because maybe that inflammation is a major trigger of progression of the disease? And then we're also interested in the vasculature, believe it or not, because the blood vessels appear to be damaged by those hits. They get leaky. We've found that in living football players, that they're, even after a single season of football, those blood vessels are leaky. And we think that leakage of into the brain of cytokines and other irritants that are normally kept out may accelerate or trigger the progression of the disease. And then lastly, the brain clears tau. In a normal situation, the brain is actually able to clear tau, along some vascular pathways. Again, you can actually get rid of abnormal tau that builds up in your brain. But we think in some individuals, those clearance pathways, those sort of garbage collectors that are most active at night, they get clogged up, they get gummed up because there's some vascular damage in this disease. And so people can't clear tau. So we think there's many different things that act together to make the disease worse.
Yeah, you're good to keep going. There's a lot of questions and you're doing a great job.
I certainly won't be able to answer all of these questions.
No, that's fine. Here's a simple yes or no. And I'm starting to work through the many questions that are coming live. Auditory hallucination. We don't see that very often, if at all, with our patients, right?
I don't remember a single case of auditory hallucinations. Generally, you think of that as, withdrawal symptoms from alcohol, maybe a mental health issue, but no, it's not in the typical repertoire.
Yeah, let's hope that means not a CT case and something treatable. Julie wants to thank and shout out to Evan Nair, who she said was uncommonly kind and immensely helpful after her husband's sudden death.
Actually, all the RAs are fantastic. I have a tremendous group of young research staff. They're extremely dedicated, but I'm delighted to hear nice things about Evan, just like I am about all of them.
Yeah, well, Julie, we're very sorry you lost your husband. There are questions about potentially some of the symptoms you may have had. Living, and again, this is a very hard and complicated question. So what is our appreciation right now or understanding of the relationship between CT staging and symptoms?
Well, that's hard. Generally, when you have higher stages of disease, stage 3, stage 4, that's when you're getting into real cognitive issues. So most of those individuals have at least cognitive impairment. They're not thinking clearly, their judgment is impaired, their executive function's impaired, or they may be demented, especially if they're in that very highest stage. Stage 1 and 2, a lot of those symptoms are behavioral, impulsivity, aggression, violent behaviors, mood behaviors, suicidality. Those are not as clear cut. We're not seeing a one-to-one relationship between the tau pathology and those behaviors, even though we know they're extremely disabling and we're extremely interested in trying to figure that out. That's where we're having a little bit more difficulty. Why are some people so erratic, so out of control, so disabled by in those early stages? Because we certainly have seen that.
Yeah, and I'll add, based on prior conversations we've had, a lot of these early stage cases, it's a complete change of personality is what the families are reporting. And what we've also learned, especially from your white matter studies, is that tau may not be the driver of that.
Right.
It may be other abnormalities that come with TBI, traumatic brain injury, and the injury and the subconcussive hits that caused this process, or other processes related to that, like the white matter damage you spoke of.
Right. Right.
So it is important to research that. We do suspect it's related to the trauma, but what exactly is driving that? We'll take work.
And actually, I just should say that we have a new grant going in this fall, specifically focused on the white matter. We're collaborating with the University of California, San Francisco. We'll be looking at some of the participants out there. But we're very excited about trying to figure out this white matter damage as a contributor to the symptoms of CTE.
Excellent. All right. Blast injury versus sports-related trauma. What do we understand about differences at this point?
It's a great question. We have seen blast people that have experienced blast with CTE. It's generally been low grades of CTE. It's not all of them. There's clearly not everyone who's exposed to blast gets CTE, but we do have a subset that has CTE. We really need more brains from these individuals who've been affected in this way to really understand it. It's a much more complicated issue than head trauma from football, because individuals who are exposed to blast, first of all, they often have a sports history. They come into the military often having been an athlete, so they already have sub-threshold injuries. Then blast injuries aren't predictable. The number, the range, the closeness to the blast, the severity of the blast, whether you're not in a Humvee or near a wall or hit the ground, all of these variables are very hard to predict and model. So we really need more experience with blast injury to be clear about it. But it's something that we're extremely interested in understanding, and we do have a grant on blast injury with the VA in collaboration with other VAs, in particular the VA in Missouri that's doing open field blast in experimental situations to truly try to focus on blast injury and what those long-term effects are.
Thank you. So someone asked, and I could potentially answer this, maybe somebody we turned down for brain donation. Why does intubation before death prevent brain donation? And I believe that's because it affects the tissue in a way that the antibodies for something like tau don't work. Is that correct?
That's right. What happens, it's a terrible situation, and I wish we could take every case, but what happens if a person is severely injured, or if they have a stroke, or if they were hypoxic, maybe didn't get enough oxygen to the brain, they can still be living, heart beating, blood pressure, but their brain is not functioning, and they may be intubated, and their life may be prolonged, but the brain itself is not functioning, it's not getting blood supply, and it deteriorates even though the person is still living. And so if there's a situation where there's been a prolonged ventilation, where the brain is not functioning, it's called brain death, we can't take that brain because we can't see anything. None of our chemical reactions work.
Yeah, thank you for answering that, and thank you for your attempt to donate. So there's a few questions on controls, which I think it's important to address. So I could probably start by saying this specific study is not one where you would use controls, because you were comparing CT cases and staging and the quantification of tau. Right. But what is going on? How do these cases compare to controls, and including how much tau is present in controls?
So there are very small amounts of tau present in normal controls, but it's an exceedingly small amount. It tends to be restricted to discrete areas of the brain, and it in no way looks like the pattern or distribution of tau in CTE. But there are very small amounts of tau in individuals who are considered normal, particularly in the locus ceruleus, and there may be mild amounts in the cortex. Where we get our controls from other studies primarily, because this study, we take people who've experienced head trauma, but we have a number of different brain banks, and we collaborate with brain banks all over the country to get controls. So we get controls from the hospital if they died from unrelated causes. We get controls from brain banks in Florida. We get controls from the PTSD brain bank. And in those controls, we do not see appreciable amounts of tau.
I do want to, a lot of people are asking about where to find help or treatment. I do want to, Julie or my team, if you guys could add the link to our helpline. So those who know us know that we have a helpline. So if you have specific questions about a referral to a local doctor who might have some experience or a willingness to expertise in CTE, feel free to reach out to us and we will answer everybody and help you the best we can with education, referrals, and mentors as well. People who've been through this or going through this and want to connect and share. There's a question on epilepsy. So somebody with uncontrolled epilepsy sent in a question. And what's your knowledge or experience with epilepsy and CTE?
Well, there are cases when people have poorly controlled epilepsy, when they hit their heads, say with a grand mal seizure, and they fall to the ground, they have repetitive seizures, uncontrolled seizures, they can develop CTE. It's not common, but it is a possible outcome of uncontrolled seizures and multiple falls. We do have 1 or 2 cases like that in the brain bank.
Okay. More questions on treatment, or actually tracking this over time. Serial DTI, what's your thought about its ability or our knowledge about its ability to be helpful?
Well, DTI is helpful. The problem right now is we're not really using it for clinical diagnosis. It's a research scan. We've done studies where we've found that DTI, which generally focuses on the axons of the white matter and their integrity, we've seen damage to the axons in DTI, not only in individuals who are living, but we've also seen them when we actually do DTI on these postmortem human samples. The problem is that it requires a lot of computational assessment. It's not the kind of scan where you go in, get the scan, come out, and somebody says, oh, it takes a long time to read. Usually the results come back in a matter of months. And right now we're not using DTI clinically at all. It's a research tool.
Okay. Inability to find words, slowed speech. Can you speak to some of the mild or some of the cognitive symptoms that people are reporting?
Well, probably the most common is, memory issues, losing your, can't remember how to get home, find yourself on familiar roads, but don't remember how to get home, losing things, having to keep lists, not recalling events, things that may have happened earlier that day or the day before, losing things around the house. Those are fairly common symptoms. It might be just poor planning, poor organization, difficulty multitasking, difficulty in thinking clearly in situations where there's a lot of stimuli or a lot of noise or other distractions.
For military folks asking about, there's a few pilots in this group where they experienced long, high G forces for a long period of time. We don't have much information on that yet, right?
No, we don't. We have very little information on that. In fact, I'm trying to recall if we have any pilots. I just don't recall them offhand. We have
No.
a parachutist who did not have CTE, but that's it.
Okay. And can you speak to, we often talk about what can you do that would potentially help mitigate symptoms or help with brain health in the long term?
Sure. The things that help with brain health in CTE are things that we all should be doing for just our general health. But if you have a healthy cardiovascular system, you're going to have a healthier brain. So physical exercise is probably the best thing you can do. Routine aerobic exercise, maintaining your cardiovascular health, it has a huge impact on the health of your brain. And there's practically no condition that doesn't improve with cardiovascular fitness. So even if you have early symptoms of Alzheimer's or Parkinson's, regular physical exercise will improve it. So that's probably number one. Then there are, the other things, watching your cholesterol, watching your fat intake, trying to eat that Mediterranean diet of high fruits and vegetables and low saturated fats. Those actually work, and it's not just your heart that you're treating. You're treating your brain. Your brain has massive amounts of blood vessels. It has the highest vascular requirements. It needs the most oxygen of all of your body, and so maintaining cardiovascular fitness is fantastic for your brain as well. And there's also evidence that it causes you to grow new nerve cells. Now, maybe those nerve cells may be in restricted areas of the brain, but it promotes a healthy brain. And then a lot of people that are early in the symptoms of CTE may be suffering from a few things. A routine, following a routine of physical exercise, structured habits, just that seems to help with just coping with the uncertainties of this disease. And it helps get you through maybe by reducing some of those disabling symptoms.
A few people have asked, how can I pledge my brain to the work? I think you're winning over a lot of people, Dr. McKee. So Julia, put that in the chat, but you can put it there again. We welcome everyone. So a little bit off topic, but I know you also just published a rebuttal as to a study on mild CTE,
Right.
and differentiating from RTAG.
Right.
Would you be interested in discussing that now in a few minutes, and we can put up the images up on the screen?
I don't have the images with me right
now. I just sent them to my team, so
okay.
if you wanted to discuss it, they could throw it up, or we could do it another time. Yeah? Sure. All right. The B-Boy to Tyler, if you got that link for me, if you're able to throw that up there. There they are. Great. So if you could scroll down, so just to explain this, this is a rebuttal to a paper that had claimed they found CTE in 6 out of 8 controls, most of whom never exposed to TBI and certainly not repetitive brain trauma.
And they were older controls.
Yeah, and you and an international consortium of CTE and RTAG experts said that maybe there was a misdiagnosis. Do you want to speak to that?
Well, just in really basic terms. So this figure that's up that you're looking at, figure 1, that's what CTE looks like. That's what CTE looks like in early stages in young people. It's a very clear abnormality. It's very clearly centered on a blood vessel. There's nothing normal about this. This is tau in nerve cells making nerve fibrillary tangles. You see how those dot-like abnormalities seem to cluster around the blood vessel. This is a lesion. It's a very clear lesion of early CTE. And then if you scroll down to figure 2, maybe superficially this looks like the same to some people, but this is, to my eye, totally different. First of all, these are veins. These are not arterioles. And the tau itself is not in nerve cells. It's in what we call astrocytes. And a lot of these lesions are found in white matter. CTE is not found in white matter. this is what we call RTAG, or has a long name, age-related tau astrogliopathy. It's something we find in old people. We're really not sure what the significance is. It might not have much significance. And of note, when you look at this paper, you'll notice that the last author is a doctor named Gabor Kovac. He's the world's expert on this disease, on RTAG. And he wrote a paper where he looked at hundreds of older people from a European brain bank. And he looked very conscientiously in exhausting detail. And he conclusively determined that none of the people in that older age group in this European cohort brain bank had early CTE. They did have RTAG, but they did not have early CTE. And so his work and the images that the people that wrote the paper that were rebutting, it's hard to say this, but we couldn't believe that paper had been accepted. It was just not accurate.
Yeah, and I just put the study into the chat so you guys can look at it yourself, or the rebuttal to the study. But it's worth mentioning, and we're wrapping up here. We'll get done here in a couple minutes. But that, as you mentioned, this wasn't found in that study of over 300 controls in the European Brain Bank. You run the Framingham Heart Study Brain Bank, right? Which is actually the largest collection of people who have been followed for their entire life and then have donated their brain. And what I know, it's been published in the Lewy body paper. How many of the Framingham Heart cases had CTE?
It's a small number. I don't want to be quoted. I don't want you to quote me on this number, but it's just a few. It may be 2, it might be 3, but some of the Framing Heart Study participants were military veterans or played contact sports. But it's out of the hundreds and hundreds of cases, we have 2 or 3.
1 or 2, yeah. It was the point is that most studies have found 1% or fewer, and with those 1%, they often find they do find that history if they are able to connect with the family of exposure. So we're getting more and more confident. I mean, there could be another way people are getting CT, but we're not finding it. Right now, a lot of the cases that have been called CT may appear have been misdiagnoses, and that's what the paper your paper points to. And I think it may be worth mentioning to the audience too, the RTAG that you were speaking of that has been mistaken has also only recently started to be characterized, right? So there's some honest mistakes may be happening here.
Right. That's the way science is. It advances, and things that you thought yesterday turn out to be not true, and or vice versa. But so this RTAG is a very new contributor to neuropathology. And as I said, this Dr. Gabor Kovac is really the world's expert in it. But in terms of clinical significance, it doesn't appear to have much clinical significance. It may just be one of those things that deteriorates over time as you age, but doesn't really contribute to any cognitive decline.
Yeah. Dr. McKee, thank you for being so generous with your hour. You still have well over 300 people here who they're writing in the chat right now how much they appreciate you. Our audience has been waiting for the great Dr. McKee to join us for a webinar. So we're thrilled to have you.
You can keep complimenting me, Chris. Keep going.
I'm recruiting you back for the next one. We've been doing 20 questions with, and so I think this group could give you more questions than we could ever expect, because this is an important issue, and your work is really advancing it. I thank you as someone who hit my head too much, and on behalf of everybody else who's joined us, thank you. Thank you to everybody for joining us, and especially our families who contributed to this research. Remember, reach out if you ever have a problem to the helpline. We're here for you. We'll hopefully recruit Dr. McKee back soon. Thank you so much to our military veterans. As you know, Project Enlist is becoming a top priority for the organization, so we can understand these differences. And with Dr. McKee at the VA, we're in a great place to advance that work. So I'll wrap it up now. Thanks for all the kind comments, everyone. We'll cut and paste them to you, Dr. McKee. I see the Stankies are here, a lot of our best friends here. Yeah.
Thank you, and I agree. Thank you, all the brain donors and other participants. We couldn't do this without you.